Hi everybody, and welcome to our module on microarrays and FISH. A DNA microarray is also called a DNA chip or sometimes a biochip. It's a solid structure that's made out of substances like glass, plastic, or silica, and it contains thousands of DNA sequences called probes that are attached to the solid structure. And it's used to test a sample of DNA that is labeled with fluorescent markers. The sample will hybridize two complementary base pairs that are bound to the solid structure, and the computer will detect which of the probes in the microarray have bound the sample, and in doing so, the microarray can identify DNA patterns in the sample. Here's a picture from the web showing you what one of these microarrays look like. Basically, these have automated the identification of DNA and made it a very rapid process because you can use this chip, see which of the probes binds your sample, and rapidly identify DNA sequences in your sample. There are many uses of DNA microarrays in research. I'll just go through a couple of examples in the next few slides. DNA microarrays can be used to identify gene expression, in other words, to determine which genes are active or inactive in a particular cell. For example, researchers may want to compare which genes are active in cancer cells compared to normal cells. This is done by collecting the mRNA from cells of interest and converting it into DNA. This DNA is called cDNA. The cDNA is then tested using the microarray, and whichever cDNA is identified by the microarray represents DNA that is being expressed in the cells of interest. Copy number variation is a phenomenon in which some cells contain increased or decreased copies of a particular gene or segment of DNA, and in some cases increased or decreased copies are linked to disease, and copy number variation can be identified by DNA microarray testing. The way this is done is that cellular DNA is collected for microarray testing and a reference sample is also tested. The computer then compares the fluorescence intensities of the sample of interest to the reference sample. So for example, if the sample has the same fluorescence intensity as the reference, this means there are no extra copies of the gene or DNA. If the sample has greater fluorescence intensity than the reference, this means there are more copies of that gene or DNA. And then if the sample has less fluorescence than the reference, this means there are fewer copies. Many genes display what's known as single nucleotide polymorphisms, or SNPs. This means that the genes exist with variations in a single nucleotide, and all the different variations can be represented in the microarray, and therefore you can identify the SNP in a particular individual gene by using microarray testing. The way this is done is, once again, cellular DNA is collected and tested using a microarray. Depending on which probes in the microarray bind the DNA, this tells you which SNP is present in the sample gene, and this can be used in many different circumstances. There are many SNPs that are associated with disease. Also, many SNPs are preserved within families, so individuals with the same SNPs are likely derived from the same family. FISH stands for fluorescence in situ hybridization, and like microarray, this technique uses fluorescent lighting to identify the binding of complementary DNA segments. In fluorescence, a fluorescent DNA probe binds to a specific gene site, and this helps to localize the gene to a particular chromosome and determine which chromosome contains the gene. So here's a picture to help you understand how this works. You take a fluorescent DNA probe that is complementary to the gene you want to identify. You then hybridize it to the DNA of interest and see where it binds. And you can actually see it light up on a particular chromosome, and this will tell you that the gene is present in that chromosome. FISH is often done on cells in metaphase. These cells have been arrested in the middle of mitosis at a point where the chromosomes are visible individually and they can be fixed to a glass slide. You then use the DNA probes that match regions of known chromosomes, and the probe will hybridize to chromosomes on the slide. This is why it's called in situ hybridization. And the probes will be visualized with fluorescence microscopy, and this will tell you which chromosomes have the genes you're looking for. FISH is often used to compare test cells to normal cells and locate the gene in the test cells and see if it's in the same place or even if it's present compared to normal cells. So in the genetics section, I have a video on microdeletion syndromes, and in these syndromes there has been a deletion of part of the chromosome. So what you will see in FISH is that the probe will bind to the normal chromosomes, but it will not bind to the chromosomes of a patient who has a microdeletion. So for example, a chromosome 22q11 deletion leads to DiGeorge syndrome, and you can identify this by doing FISH, and you will see that the FISH probes will not bind normally because there has been deletion of a genetic segment. You can also use FISH to identify translocations. It will show fluorescence on the wrong chromosome if a gene has been translocated. You can also use FISH to identify duplications. If a gene has been duplicated, you will see an extra site of fluorescence compared to normal cells. And that concludes our video on microarrays and FISH.